Hello, my name is Martin Blaser. I'm the director of the Center for Advanced Biotechnology and Medicine at Rutgers University. And today it's my pleasure to interview one of our new faculty members at CABM, Dr. Michael Manhart. Dr. Manhart came here not long ago, and he's an assistant professor in the Department of Biochemistry and Molecular Biology. So welcome, Michael, and maybe we can just start. Tell us about your early education and what you were doing before you came to CABM.
Yeah, maybe I can take one step even a little earlier than that and say that for me, kind of what got me really interested in science, I think as a child was, really kind of more my interests in math. And a lot of that was really about my interest in using abstract concepts to describe the world quantitatively and to identify general patterns. And so during my bachelor's degree, that got me interested in studying physics as well as math. And then it was at the beginning of my PhD where I started getting interested in biology because I saw it as this place where we could apply the same kind of reasoning, looking for these general quantitative patterns in this, extremely complex, body of phenomena that I would say we really know very little about. And so I did my PhD in biophysics here at Rutgers. And then after that, I was a postdoc studying biophysics and molecular evolution at Harvard University. And then after that, which was just before I came here, I was a junior group leader at ETH Zurich in Switzerland, where my group started to really focus on microbial ecology and evolution.
Great, great. So it brings us from childhood to the present. So how long have you been here at CABM? And, what's the main focus of your work right now?
So I've been here at CABM for 5 months. So I started here in January of 2023. And the main focus of our lab here is quantitative evolutionary microbiology. And I like to kind of break that down into these 3 words. So we focus on microbes because, of course, they impact human health in so many ways, but also they underlie every ecosystem on Earth. And we focus on the evolution of them because that's really always the organizing principle of every aspect of microbes that we observe. And then finally, we take a quantitative approach to these systems because I think these numbers really matter. And so I like to think of the example where if you had a bacterial infection and you knew it was going to evolve resistance to an antibiotic, you'd really care if that was going to evolve resistance in 2 days or 2 years. And so we really try to focus a lot on trying to understand these quantitative aspects of microbes like that. So I think the simple version of kind of what we do is we try to understand how microbes grow, interact, and ultimately evolve. And I think a more complex version of putting this is saying that we want to understand how fundamental evolutionary processes like mutation and selection both shape and are shaped by the ecology and cellular physiology of microbial communities. And we especially want to explore this in biomedical contexts like antibiotic resistance and infections caused by multiple microbial species.
So that's a pretty big universe. What would you say is, 1 or 2 of the key questions that you're interested in right now?
So there's kind of been 2 major areas of work in the last few years. One has been focusing on understanding the population dynamics of microbes, so basically how they grow and what that means for their ecology and evolution. And there, one of the things we're really interested in is understanding to what extent microbes are growing more or less at steady states, where they're just kind of in some, constant state of growth for long periods of time, versus to what extent they're undergoing cycles where they're growing a little bit, then switching to a different state of growth and maybe dying. And there's constantly these transitions happening between different states. The other area of work that's a little more recent for our lab is trying to understand how interactions affect evolution of microbial communities, and in particular, whether interactions tend to make adaptation to a new environment happen more quickly or more slowly.
Yeah, great. So how are you approaching these questions?
So in our group, we use a combination of laboratory experiments and also mathematical modeling. And we focus quite a lot in particular on, I would say, synthesis of these two things, which is what I think of as really developing theory. So we try to connect many different experiments, both ones that we do in our lab, as well as other ones from other people's work, and then models into sort of a coherent framework. And kind of the way we try to do this is we usually focus on sort of simplified systems that we can study in the lab or through these mathematical models, where we can really precisely control many factors to understand some general principles. And then our goal is to use this to generate hypotheses. And then we search for ways to test for those hypotheses in natural systems.
That sounds good. Any particular project that is of great excitement to you right now?
Yeah, so the second area of work that I mentioned, understanding how interactions affect adaptation of communities, is, I would say, one area that I'm really excited about because it's the newest in the lab, and we're kind of developing a lot of new methods for it, which I like. In particular here, we're focused on understanding how interactions affects mutations. And so, for instance, does the effect of a mutation change when you have microbes that are interacting with other species compared to in the absence of those interactions? And so what we're doing for this is taking this very direct experimental approach, which also I like. We're just literally generating enormous collections of hundreds of thousands of mutations in bacteria and testing the effects of these mutations in the presence and absence of these interactions. And one of the aspects of this that is kind of, I think, going to be important in the next couple of years, especially here at CABM, is understanding the role of spatial structure, which is something that we know, of course, microbes live, in these spatially structured environments, in the human body and everywhere in nature, but we know very little about that. But of course, it has to affect a lot their interactions because, who interacts with who is, of course, determined by who's next to who. And so this is going to be an important theme, I think, going forward.
Right. I'm excited already to hear about the results. Now tell us, what school are you in at Rutgers and what department?
So I'm in the Robert Wood Johnson Medical School, and my department is Biochemistry and Molecular Biology. I also have a secondary appointment in the Department of Medicine.
Right. I think in the Division of Infectious Diseases. So
That's correct.
yeah, I think that makes very good sense. So now tell us, how do you interact with students and trainees and people in your lab?
So for me, the key word is, I guess, kind of like in our research, adaptation, because I think different people in the group need different things. And I think those things also can change over time as, people go through the ups and downs of projects. And so I really try to avoid having too much of a one-size-fits-all model to how I work with people in the lab, but really always try to be ready to adapt to these different situations and really try to kind of cultivate, a culture of transparency and communication with people so that people feel like they can tell me very explicitly what they need to be successful. And I would say another thing that I really try to work on in our group is cultivating sort of collective behaviors of our group as well, and avoiding a situation where people just interact with me, but rather try to make sure that, we take advantage of the collective strengths of the different people in the group. There's some things that I am good at, but then there's other things that other people in the group are better at. And I think this is something where the group really leverages its power. So I really try to kind of find ways to cultivate that in how I interact with people.
Yeah, great. So do you have any other roles at Rutgers or in the scientific community in general or the world at large that you want to tell us about?
Yeah, so kind of as a reflection of the interdisciplinary nature of my own background and what our group does, I have been getting involved in many other programs here at Rutgers, in particular the Rutgers University Microbiome Program, of which you are also a leading figure. That has been a major source of interaction for me. And I've also been cultivating relationships with another number of other departments at Rutgers through graduate programs. So this includes molecular biosciences, as well as microbial biology, ecology and evolution, and physics. And so this has been something I've really enjoyed and found really amazing here at Rutgers, that it's such a big and diverse place. And it's been very easy to make these kinds of connections across the university. And for me, this is just essential for the kind of science we do.
Great. Okay, so I have just one last question, and that is, it's a little unusual question because you're like a young person. So the question is, let's say it's the end of your career and you're looking back at your career, and we had to write some kind of scientific eulogy to you or obituary. What would you want to be remembered for?
Yeah, I think it's a great question actually to think about at the beginning of one's career, to kind of focus one's goals. So scientifically, I like to think about what the textbooks for our field would look like in the future, because I think textbooks usually kind of reflect the most canonical established knowledge for a field. And so I think my hope for, microbiology, say, would be that the textbooks for the field in the future would be organized more around sort of major concepts, things that are these fundamental principles in microbes, like growth, resource consumption, interactions, and of course evolution, and having each of those really, intimately tied to quantitative data, sort of fundamental experiments and models, and to structure it that way rather sort of than the way that I think has been traditionally done a lot of microbiology, where we think more in terms of breaking things down in terms of the constituent molecular and cellular systems. And so my hope would be that the contributions of my career will be towards establishing what some of those major concepts are, and what are some of the key experiments and the key models that we can have used to support what those are. And of course, in particular, I hope that we would be able to find ways to connect some of those fundamental concepts to the role of microbes in human health. So for instance, finding something about how microbes evolve that would tell us how a disease works, or a strategy we might be able to use for treating a disease. And one thing I would also say when I think about my career is that I think I'm completely okay with being wrong about many things, because I think my goal would be, as long as I can contribute things that will help propel the scientific community forward towards being right, that's what really matters. And so even I think sometimes being wrong can be a very valuable contribution if that incentivizes other people to find what's actually right. And so even if that turns out to be the case for some of the things that I do, for me, I think that would still be success if it still contributes towards the community as a whole finding the correct knowledge in the future.
Yeah, very well said. I think that anytime you test a hypothesis, you can reach a definitive answer. Whether it is correct or incorrect, you improve the knowledge of the world. So, yeah, very good. You're old beyond your years. So, okay, great, Michael. Well, that finishes all my questions. Any other questions you want to bring up? Any other subjects?
One other thing I just thought about, kind of related to this theme, is I think in a lot of areas of science, including our own of biology, people sometimes ask whether we've reached the end of theory, due to the explosion of data and the usage of machine learning type methods. And I think it's worth pointing out, at least in my opinion, that I think a lot of those same observations actually reveal just how essential theory really is. And one of the things that I've really learned about this question, having started to work so much in the field of microbiome research in the last several years, is there are so many places in this field where there is a ton of data, but there's just so little understanding about that data. And I think for me, we're actually in a critical time for biology as a whole, but especially microbiology, where we really need to develop a lot more fundamental theory. And I think this is going to really be kind of the next generation of our field. And so that's something that I'm really excited about. and hope that I can contribute to in my career.
Yeah, that's also very well said, and I agree with you. Theory is not dead. I don't think it will ever be dead. So the needs are there. Well, thank you. Thanks so much. And I think we're all done. Bye.
Great. Thanks so much, Marty.